Expression of a cloning vector. Here the yellow is one is the insert having a threaded stop codon, and here's the vector having a multiple cloning site within the leg jet gene and a promoter. When it get inserted and get transmitted into your bacteria for cloning and to express that target protein. During this transcription process, the stop codon helps to prevent during this translation to make protein with only a fragment of this NH2 and COH. This producing a fusion protein with the undesired fragment is not being translated here. This can be controlled or regulated by using very number different type of promoter. For example, if you use the leg open promoter, as it is not that a strong promoter, so it will not be going to helpful to produce huge amount of protein of interest. Using Arabinose promoter like this one shown here shows that this amount of the expression of the protein is much more enhanced with the increasing concentration of this inducer. In this picture you can see here that is a tryptophan operon promoter with your histidine in its coding gene and a site where the enterokinase could recognize to cut it the protein that will form, and next to it is your cloning site, multiple cloning site. So when the bacteria produces protein, that cell must be lysed to release all the proteins. The chromatography technique being used here is your affinity one. This affinity chromatography binds specific to that histidine to that of your nickel, and rest are eluted. With that nickel column containing that target protein that can be used this histidine imidazolam to release. Once this protein is released with the help of enzyme enterokinase, we can break this bond to get the target protein. Once this get done, we do that again to elute the target protein, leaving this histidine hexadine intact with that of your nickel. That is your oligohistidine, which is a 6 encoding histidines together that have affinity for that nickel. So this is how we get a target protein from this expression vector